of Chungnam National University. In this presentation, I'm going to talk about the taxonomy review of the tribe DRABESCNI from Korea. Contents in the following order. South-American DELTOCEPHALINAE was described worldwide in Europe or territorial habitats in the largest subfamily of CICADELLIDAE based on the number of described species. Most of them include species that are so similar they are difficult to distinguish. They feed on plants and some are economically important plant pests. Because they damage plants directly by feeding and ovipositing, or indirectly by transmitting plant pathogens. In previous research, two groups previously formally DELTOCEPHALINAE, DRABESCNI and Planthoppers, were included as the subtribe of DRABESCNI. The tribe DRABESCNI contains two subtribes, namely DRABESCNI, that contain the Drabescus and the Lengatilla. And the larger subtribe, the Planthoppers, contains more than 36 genera. They are described in the upper tropical, Australian, Oriental, and Palearctic region. There are 240 species recorded worldwide, with only 14 species recorded in Korea. Since 2007, the National Institute of Biological Research, or NIBR, in South Korea, has diligently compiled on the annual list of Korean records for species. According to this year list by NIBR, a total of 14 species, 8 genera, and 2 subtribes have been recorded in Korea. The tribe was first recorded in 1932 by Boy, who described the Drabescus librigenotus from Korean Chungnam. Baron Doi, Nagaoka, and last report the Drabescus fasciatus, Drabescus openai, Artisanosis thalicis. Then Korean researcher Lee, Won, and Ho conducted further active study and reported 9 more species. A brief introduction to the morphological feature of tribe. They are sharing the following characters. Both group of DRABESCNI have been with a long appendage on the pilocar that do not persist into the other stage. Placement of an antenna has height on the base, and antenna elongate about 1 to 1.5 times width of head or longer. Here, the morphological picture of the two tribes, DRABESCNI on the left, is relatively wedge-shaped and has the dark body colors such as brown, dark, and gray. Planthoppers are slender and bright yellow, green to brown. Also, it can be seen that DRABESCNI has a strong antennal edge, whereas Planthoppers has a weak or absent antennal edge. And if looking at the wing, in DRABESCNI, the appendix is broad and extends to wing apex, whereas Planthoppers is not broad. For this research, we have collected literature like as the catalog and original description, including the Korean fauna. Samples were collected by the sweeping, light trap, and Malay trap. In addition to specimen held in laboratory, we identified specimen held on domestic institutions. We also checked the material collection at Hokkaido University. We saw some type specimen for the review of Korean record species. Specimens were taken under light and stereo microscope and digital camera. At Methanoid removed from the specimen and soak in 10% potassium hydroxide at the 17 degree to dissolve and wash in the distilled water. And then the transfer to glycerin for further dissection and examination. Let's turn to result and discussion. We found two new liquor species of the genus DRABESCNI. These are the species DRABESCNI spontanus on the left and DRABESCNI spurgatus on the other. DRABESCNI spontanus is previously distributed in China, Taiwan, and Japan, and we confirmed the distribution in Korea based on a sample from Jeju Island. And the species DRABESCNI spurgatus was described only from China. In our study, we reported in Korea for the first time. Previously, only one species of genus Batia has been recorded in Korea, but we newly identified one new liquor species of the genus Batia and one species of the genus Batianus, which are morphologically similar to genus Batia. In Batia sazenensis, the Edelus and Bicolor were different from the Batia koreana, and we newly checked the distribution of Korea. The genus Batianus is characterized by the one pair of process, so the inner tube is well sclerosed. This species, the new species, differ from the two species in the genus Batianus due to morphological differences in the EDR effects. In 2022, we reported the Auchenorrhyncha pallida for the first time in Korea, although Daejeon was the reported post in previous research. We obtained a large number of nymph and others collected over several years of street plant Auchenorrhyncha indica or creep micro in South Korea, and present new host plant information for this species. In our study, we referred the genus Planthoppers based on the new description from the Korean fauna. After this genus, after original description, 4 new species had been described in China, and 2 species at the Daejeon and Chang from Sabah, Malaysia. In this year, we refer to newly recorded species of the genus Parabolophona from Korea. The previous described species, Parabolophona guttatus, is described in the southern region of Korea, where the host plant cover tree is native. Parabolophona guttatus has a different distribution from the other 2 newly recorded species. Planthoppers Ishihara and Planthoppers due to the distribution of host plants and is clearly distinguished by the morphological differences in male genital structure. The genus Vigar was previously described in China and Japan, where only Vigar bonensis was recorded. We have collected 2 morphotypes in Korea, one of which we reported in 2021 as a new species hosting the air carpet tree, along with the type specimen of Vigar bonensis. In the subtribe DRABESCNI in Korea, only the genus DRABESCNI has been recorded. The 6 species were previously recorded. Our study confirmed the domestic distribution of the 5 species except the DRABESCNI lymphaticus. We found 2 new liquor species and reflected liquors with the current classification. Until now, the 5 species and 5 genera of the subtribe Parabolophona have been recorded occurred in Korea. In our study, we referred the distribution of the four new records and three new species from Korea. In our study, we also update the record of the Korean Drabescus species. Firstly, the species of record as Drabescus conspicus in the NIDR was made a senior synonym of the Drabescus lymphaticus by Zhang and Wei in 1996. And Drabescus conspicus was collected to junior synonym of Drabescus lymphaticus in 2012 by the Zhang and 2019 by Lu et al. Therefore, we adapt the Drabescus lymphaticus from Korean Baños. In addition, Drabescus hematiformis was treated as Drabescus nigripematus and junior synonym by the Zhang in 1996, and we also update this in Korean records. Also, according to Zhang and Wei, some records of Drabescus nigripematus are domiciled to case by the Vivaste in 1971. The encrypted and Eunji and Lee. The misidentified species was described as the new species Drabescus vivaste in 1996. Therefore, some of the records of Drabescus nigripematus in Korea are also the Drabescus vivaste and should be reflected in Korean record. Here, our citation, namely that the reference in Enuprib in 1971 not vivaste. In summary, in Drabescni, two new record species were referred for the genus Drabescus from Korea. Two species, Drabescus conspicus and Drabescus hematiformis, were revised to Drabescus lymphaticus and Drabescus nigripematus, respectively, and Korean record of Drabescus vivaste was updated. We conducted a genital experiment of our Korean Drabescus species except Drabescus lymphaticus. In Paraboloponina, we obtained a sample for our species referred in Korea and conducted genital experiments, with the result in the identification of additional species. In this study, we combined the total of 21 species, 10 genera into subtribe from Korean fauna, including 3 new species, 6 newly recorded species, and to update Korean records. Finally, we provide the revised checklist of the Korean Drabescni. This is reference for this presentation. We are grateful to Professor Kazunori Ishihara in Kaido University for access to precious specimen, and we also thanks to Juni Group for allowing us great picture. This was supported by the grant from NIBR in South Korea.